Okay, so we're now going to do cytology, which is the study of cells, not tissues, cells. And it's about the cell and allowing things to come in and out of the cell, and also what's going on inside the cell. And the best way I could really explain what's going on is an analogy of the cell with a village with a castle. Okay? Around the village, the castle, so we have this castle over here, right? So there's a castle, there's some other houses and other things in their little shops, a little village. The castle's the main thing, as much as what we're going to learn is the nucleus is the main thing. And then we have this moat of water going around the whole area, kind of protecting, allowing certain things, as the drawbridge comes down, it allows certain things to come into the cell and certain things to go out of the cell. If the drawbridge is up, it's not going to allow anything in there. And we're going to learn that a plant cell even has more things further around the moat, like maybe a whole forest around there, that is really going to protect it from other things. Okay? But not all cells have that. Plant cells have that cell wall, but animal cells do not. You're going to find that there's roads in here. That'll be our endoplasmic reticulum. You're going to find that there's little shops in there, like the wood shop and the metal shop, that's going to make things so that they can either get it out of the cell to sell it to other villages or to be used inside there. Those are our ribosomes. So we're going to talk about this castle and this moat and draw this analogy of how it looks with the cell. All right? Now I kind of split it up. Today what we're going to talk about is the membrane and how things go in and out of the cell, called transport mechanisms. Tomorrow we're going to spend a lot of time just talking about the parts inside the cell, and then the next day we'll talk about DNA and kind of like the constitution or the blueprint of that cell and how to make a copy of it. And then we'll get into how mitochondria works, and then we'll talk about how to make a protein and genetics, and then we'll be done. All right? So that's where we're going with this. All right? Questions about where we're going? Okay? All right. So let's get started with it. All right? Simple things. The function of the cell. All right? Just pretty straightforward kind of information. I'm not going to ask questions about this per se, but it gives you some introduction. It's the basic unit of life. It's there to protect and support. All right. Movement. There's various kinds that will allow the cells to move. All right. Specialized ones. You'll see things like the sperm is one cell that can move. It has a little flagellum. Okay. We also have communication, where one cell can communicate with another cell. There's cell metabolism and energy release. That's a whole separate test. All right, we'll talk about that later. And of course, inheritance, that you could pass on this gene or this cell to the next generation and so forth. All right, so we'll talk about DNA with that. All right, so this is our run-of-the-mill type of cell. It has different parts to it. Into those later. But you've got the membrane going around this whole thing, allows certain things to go in, certain things to go out. You'll see a big nucleus there that kind of tells everybody what to do. Think like this is the United States, Washington, D.C. is our nucleus, right? We got mitochondria over here that are actually going to be the powerhouse, makes the actual ATP, and a slew of other things that'll do make proteins, or there's things in there that are going to protect the cell, like little guards, lysosomes. We'll talk about those later. A cell can look like that. A cell can have different shapes and sizes. Okay? I mean, this is more like a round one. This is more like a trapezoid. But we can have cells that are very flat. There's one cell there. squamous cell. They could be like a stop sign, hexagon shape. They could be square, cuboid. They could be long. They could be spherical balls going around there. They could look like Cert's candies, okay? Or Smarties candies, right? They could be star-like. They could be football shaped, called fusiform. They could be very long. I mean, your nerve cell is literally this long. I mean, it's going to go, let's say, from my finger here all the way to my spinal cord. And sometimes it even goes into the spinal cord and up to the brain. So they can be really long, very thin. You can't see them with the naked eye. But some of these are long, some of them are small. So you can't say there's a typical cell. They're all different sizes and shapes. Okay? So we have this dividing structure. And we talked about this before. Alright? So intracellular fluid is fluid inside the cell. Extracellular fluid is fluid outside the cell. We abbreviate it with ICF versus ECF. So we have stuff inside the village and stuff outside the village. Keep that in mind. This membrane is semi-permeable, which means it's going to allow certain things to come in and certain things to go out. We want the men to leave the village, the knights leave the village to go fight the dragons. We don't want the babies to go out, right? It's going to allow certain people to leave. We want things to come in. People are trying to sell things, raw material to build buildings in there. So we need people bringing in wood into the village. We don't need burglars coming in. So it allows certain things to come in and certain things going out. Now, plant cells have that special thing called a cell wall. All right? So a plant cell, and this is also found in prokaryotes, fungi, and some protists, they have a cell wall in addition to the cell membrane. It has both, not one or the other. Okay? A cell wall is much, much thicker than the cell membrane. Again, think of the cell membrane as the moat around the village or the castle area, but then the thick forest, and it get really thick, is going to be around the moat. An animal cell just has the moat. There's no forest around there. So yes, you're already probably thinking pros and cons about this. Some advantages, some disadvantages about a cell wall. So cell walls, I'm going to make it brief for you, but cell walls in plants, they're made up of, MUO is made up of cellulose fibers. Remember that polysaccharide that you put in your diet, but it's more like roughage? Right? It's not starch, the stuff that you actually get energy from. So you need the roughage, the fiber, that's what the cellulose is. So the cell membrane, or cell wall, is made up of carbohydrates. See, that's where, and again, I'm not talking about the test, but this is where you're going to have to put stuff together. All right? I don't say that the cell wall is made of carbohydrates. We learned about carbohydrates. You learned about that in chapter 5. The carbohydrates, polysaccharides, make up cellulose. But maybe on the test, I might say that the cell wall is made up of polysaccharides. I won't say the word cellulose. See, I find that many students just memorize. Wrong. You may work in high school, but it won't work in college. You need to be able to apply this stuff. If I said what type of biologic macromolecule is making up the cell wall, there's nothing wrong with saying a certain type of polysaccharide. Sugar. Don't just memorize cellulose, because you're going to forget a lot of stuff. You've got to be able to apply this stuff. Does that make sense? Okay? So that's what the cell wall is made of. There's sugars in there and other proteins. It's much thicker than the cell membrane, and it helps protect the plant cell. All right? Keeps the shape of the cell also. Since it's so rigid, it's hard to change the cell shape on there. And it prevents excessive water uptake. So it prevents excessive water to go inside the cell. All right? So plant cell walls, we have multiple layers. The trees, whether you have tall trees in one layer, smaller trees in another layer, and shrubs in another layer, right? Think of it like that. I'm trying to wire your brain to come up with these analogies into your head. It takes me longer for me to explain all those things, whether shrubbery or tall trees, but in my head, I can come up with this much faster. And that's what you're going to try and do as you go through course after course after course, and come up with analogies in your head much easier and faster than explaining them. So you have this primary cell wall that's relatively thin and flexible. A middle lamina, all right, or lamellae, rather, is another layer, and that's a thin layer between the primary wall and the adjacent walls. And I'll show you a picture of this. We also have in some cells a secondary cell wall, all right, and it's added between the plasma membrane and the primary. You also have plasmodesmata, and that channel, those are channels to communicate from one cell to another in plants. There's other kind of connections between animal cells, things called desmosomes and hemidesmosomes and stuff. You'll learn about those another time, but in plants we have these. So what you got here, this is one plant cell, here's another plant cell. You got these little channels that connect between them. Those are the plasmodesmata. Okay. You also see the thick plant cell wall, and the yellow thin line is going to be the cell membrane itself. Okay. So it's just a very schematic view of what this is. But keep in mind, that if we get a close up of the cell wall, you'll see it over here, and then trying to show you those different layers that I just mentioned to you. Okay. Think of the forest around the moat. And different kinds of trees, different kinds of layers. But now let's direct our attention to the cell membrane, which all eukaryotic cells have. Okay? The cell membrane, also known as the plasma membrane, has what we call a membrane potential. Now, I'm not going to bore you with a lot of this. You'll learn about this in other courses. But understand what a membrane potential is. We learned about charges. We have cations, anions making charges. And a membrane potential is that if there's an equal number positive outside and an equal number of negative on the inside, then the membrane potential is going to be 0. Does that make sense? None of the things want to go on either side because we're at a neutral spot. That's not the way it works, though. We want things to go in, something to push in. If there's a voltage that actually if there's more positive on the outside, that positive wants to come inside. Is that a concentration or a voltage gradient, right? So you want to have that. Otherwise, the cell will be dead. There's nothing that will go in there. So we call this a membrane potential. Why is it called potential? Because it's the potential to go to 0. It never goes there, because if it did, you'd be dead. So there's something, if positive is going to come in, then there's going to be a mechanism to try and push it out so that we can actually have this constant gradient coming into the cell or out of the cell. Okay? So we call this membrane potential. And we have more positive in a normal situation. There's more positive or cations outside the cell than there is on the inside. So the inside is more negative, right? So we also have on this cell membrane is something called glycocalyx. So you're thinking glyco, right? Think about that. Glyco, that means sugar, right? Glucose. So there's parts of here that are carbohydrates. And these are cell markers on the cells. And some of them could be carbohydrates and proteins, some would be carbohydrates and lipids. We have glycoproteins and glycolipids. And these are little markers. I think I used this scenario once before. If I tell you how to get to my house, and every house on the street is green with a white picket fence, and I say, oh, my house is the green one with the white picket fence. You don't know which one it is unless you put a marker on it. A number 22, that's a marker. A pine tree, that's a marker. A windmill, that's a marker. Changing it to red instead of green, that's a marker. So this helps to determine one cell from the other, to help it so that other cells or other communicable substances can say, all right, I could recognize that cell because it's red or something, right? So that's what that is. Glycocalyx are little markers to help other things recognize what that cell is. What is that? That's a phospholipid, right? We talked about that. See how the chemistry's coming back over here. So that's a phospholipid. And we say that it's an amphipathic particle. That's a new word, but we've been using it, but I haven't used the term though. Amphipathic means that there is a polar and a nonpolar end to it. A positive and a negative. And we talked about that the head is hydrophilic because it's polar, and the two tails are hydrophobic because it's nonpolar, right? This is where the fatty stuff would be. That's where the charge would be. And we kind of drew a picture of what it would actually turn into if we put them all together, right? It turns into this situation. So look what this would be called. You got two layers. So that's a phospholipid layer, and that's also a phospholipid layer. So we call it a phospholipid bilayer. Tell that to your parents. Oh, I just learned about phospholipid bilayers in the school. Like, we'll just continue going. What's a phospholipid bilayer? Oh, it's a membrane around the cell. Well, why didn't you just say that? Because I'm a college student. Right? It's the same thing. All right? In fact, when I was teaching my daughter some words to use, and she said, oh, I got this trolley horse. I said, that's not a trolley horse. That's an involuntary contraction of skeletal muscle. And they're like, now she uses it in school. I had an involuntary contraction of skeletal muscle. And they're like, they don't know what they're talking about. It means cramp. It's the same thing. All right. All right. So this is how it would be. I kind of talked about this. If we had like a beaker of water and there was air there, they would form a situation like that. Okay? But that's not the way our situation is. It turns into this, but think of it as a fluid mosaic pattern where it's not just one layer, you've got to think three-dimensionally. Okay? And it's fluid mosaic, fluid because it does go like this. It's flimsy. We're going to learn that there's things that go inside this membrane that'll make it a little bit more rigid. We'll talk about that. There's other things in here, proteins. You see, this allows certain things to come into the cell, certain things going out of the cell. But certain things can't get through there are polar things, things with charges. They can't get through here. They have to go through tunnels. And that's what some of the proteins are doing. They're making these little tunnels. Okay? Some of them are gated, have gates on them, some of them don't. So, going backwards, here's your phospholipid. Turns into a phospholipid bilayer. You put more proteins in there, you got the cell membrane, and there's the whole cell with this phospholipid bilayer around it. Okay? If we put it together, if there's air there, it would form this way. But normally, this is what it'll turn into if there's no air, right? And inside your cells, that's what that is. It's a phospholipid bilayer that is dividing two sets of fluid Inside is intracellular fluid. Outside is extracellular fluid. We got to get some stuff going from the outside to the inside. This cell membrane is going to allow that. Well, certain things. And vice versa, there's certain things that need to go from the inside and go out, and that cell membrane is going to control that also. We've got to talk about those. It's called transport mechanisms. Okay? So again, don't worry about a micelle, you worry about them in the digestive system. But it forms this kind of thing. All right, they call it a liposome, but if you think about a three-dimensional thing, that's what's happening here. So you have things inside and outside the cell. And there's your bilayer sheet. All right, this is close up to the same thing. So what else is in this membrane besides the phospholipid bilayer? Okay, cholesterol. Yes. You need cholesterol for every cell in your body. Now, you don't need an absorbent amount of cholesterol. In fact, your body makes its own cholesterol too. We call that endogenous cholesterol. There is cholesterol in foods, and we call that exogenous cholesterol. Cholesterol comes from outside the body and it's going to come in. You don't need the exogenous cholesterol because your body can make cholesterol. But cholesterol tastes so damn good, though. And that's why we got to put cholesterol in food so you can enjoy the food, because you got to get that food in your system because that's glucose, or it will turn into glucose, and you can get energy. So we got to make something that's enticing, right? You got to make something that's going to taste good and put it in there. Well, cholesterol is in the cell membrane, and that's what's going to give it rigidity. In other words, it normally would be like this, but cholesterol in there is going to make it go like this. So it gives it some rigidity. Does that make sense? All right? About 20% of the cell wall, 20% of the cell membrane is going to be made up of cholesterol. We also have proteins in there too, and there's two types of proteins. We can have integral proteins that go through the whole span of the cell membrane, from top to bottom. Okay? And most of those will be those channels I was talking about. But we also have some that are peripheral proteins that will either be just stuck on the top of it or stuck on underneath it. Okay? Some of them are used as markers, receptors, attachment sites for things. So we have here one that's going right through here. That's an integral protein. Sometimes they can have a tunnel in there, allow certain things to come through and certain things to go out. Other proteins could be just on one side, that's a peripheral protein. Or there could be one over here. All right? And this is putting it all together. There's your phospholipid bilayer. You can see the integral proteins. You'll also see some of the peripheral proteins. The yellow that's in here is the cholesterol that's scattered throughout there to give that cell membrane some rigidity. Okay? Questions so far? All right, so let's talk about these proteins that are embedded in the membrane. And we have 6 functions of these that you need to know. So now we're going to get more into physiology or function. Before, it was all anatomy, right? It's all about how the structure is. But now let's talk about what they do. So very briefly about these. One of the things it does is that we have receptor molecules, all right? Something's going to go on it. Like a keyhole is on a door. So the key is some kind of we call it a ligand, or some kind of substance that needs to go into a receptor. So some of these proteins that are embedded in the cell membrane are used as receptors. Okay? And it's just this kind of picture here. There's the receptor protein, some kind of ligand. Ligand is anything that binds to the receptor. Will just bind to that. All right? Like a lock and key. All right? Sometimes you could have a combination going on. You could have a protein that acts as a receptor, but it's also a channel. Right? So you have this protein here. There's receptors on here that something's going to go on, and if they go on, then it might open up a gate to allow certain things to go into it. Right? To open up the gate to get into your house, you might have to have a receptor in there, or I'm sorry, a key to unlock the receptor, and then it opens up. Other times you might just have to push something in there and it does something inside the house. Maybe it turns on the lights. What I'm saying is certain proteins might be a receptor and it might be a tunnel. Some of them, and I don't want to go into heavy with this, are G proteins. Basically, it's a receptor that goes on here, and then this protein changes shape to allow other molecules inside the membrane to do something else. It's like a chain of reactions. It's pretty heavy. I don't want to go into heavy to talk about it much, but it's kind of like what goes on inside the keyhole. I mean, all you do is put a key in there, turn it, and the door opens. But there's something going on inside the keyhole. Does that make sense? That could be going on. All right? So we can say there's a G protein. Some of these protein molecules in the cell membrane are enzymes. So there could be, let's say, some kind of dipeptide there. It goes on top of here. This actually is an enzyme that will break it up into 2 amino acids or whatever. Okay, does that make sense? All right, so some of them could be enzymes. Some of them could be carrier proteins. So what's happening here is that this structure, whatever this is, molecule, looks like a star, it can go through here, but it has to be able to fit in there. Like a glove, like a snug glove, right? It has to fit in there. And if it does, then it can actually move it right into the cell. But if it was round, you wouldn't be able to do that. So certain channels are only going to allow certain things to go through. We have non-gated channels. All right? These are always open. And they're going to allow certain things to go through that have a charge. So if we have more sodium on the outside, well, that's not a good example. If we have certain cations on the outside, and there's more of it out there, and there's less inside, as long as that gate's open, which is in here, it's just going to go through from a high concentration to a low concentration. Okay? So those are ions that are doing that. We also have gated channels. As the name implies, there's some kind of gate there, and it is a gate. Certain things might, just a certain charge might open it, or certain keyholes. I mean, like something might have to attach to it, or certain things, it's just electricity. And it's basically, we have 2 types of doors, right? You put your key in the keyhole, and then manually we'll open it. And there's other doors, you just go in the area, and it's electronically, it senses someone's there, and you can walk right through it, right? Or sometimes there's a keypad on it. So we have different types of things that open up doors or gates. Okay? So we get a ligand gated, which is, again, that's more like a key and keyhole. Right? Something has to be bound to the receptor over there. Or we can have voltage gated. You have a keypad. It's not a keyhole, it's a keypad. You got to get the right combination to cause the electricity to happen to open up the door. The marker molecules, as I mentioned before, right? It allows other cells to identify one or the other. Well, those are only going to be found on the outside of the cells, because inside it already knows it. So it's going to help on the outside. All right? And it'll help to determine if the cells, or whatever that thing wants to get in, if it's friend or foe, right? Something that it wants, friendly, or something that's going to be like an enemy. And we also have the attachment sites. So proteins over here help to attach things. Kind of like the studs in a wall, so that we could attach sheetrock to it, right? That's what, so think of the proteins in this case is the studs in the walls.